I am Dr. Silvan, Assistant Professor, PG and Research Department of Biochemistry, St.Joseph College of Arts and Science, Autonomous, Cuddalore. I am going to present the topic DNA and repair mechanism. We will discuss about the two major repair mechanisms, namely photoreactivation and excision repair. DNA is easily damaged under normal physiological conditions. The return of damaged DNA to its normal sequence and structure is called repair. Many different kinds of physical and chemical agents to damage the DNA. Some of these are endogenous agents and exogenous agents. Cells usually can survive DNA damage provided that the replication and transcription machinery can still perform the functions. Biochemical mechanism for repair of thymine dimers. There are four major pathways dealing with thymine dimer in DNA. The first one, light dependent repair and light independent pathways. The three other distinct mechanisms are excision repair, recombination repair and SOS repair. The first repair mechanism, photoreactivation. First, the enzyme cleavage of thymine dimer is activated by visible light approximately 300 to 600 nanometer. The enzyme is called PR enzyme or photolyase. The light energy cleave the C double bond C bond of the cyclobutyl rings. The first enzyme bind specifically to the thymine dimer. If light absorbed, the thymine dimer monomerized. Finally, the photolyase dissociates from the normal pair of thymidine. The process of photoreactivation, initiation, repairing and release the enzyme. The first initiation, photolyase enzyme bind to the DNA and creates enzyme DNA complex. Then the repairing process, when the absorption of visible light and activation of the enzyme, and then the thymine dimer is monomerized. The final, the release. After repairing the release of enzyme, the thymine dimer was monomerized. The DNA damaged strand was completely repaired. The second repair mechanism is excision repair. The excision repair essentially consists of removal of a segment of DNA containing the damaged portion of one strand of DNA and new synthesis of the removal segment of the DNA strand using the undamaged strand as template. The first step is known as excision steps, involve recognition of the damaged segment by an endonuclease. The enzyme cleaves the phosphodiester bond of the sugar phosphate backbone at the 5' end of 8 nucleotides ahead of the damaged site, producing a 3' hydroxyl group was free. The next step is known as excision steps, involves a cut at a site 4 to 5 nucleotides downstream from the damaged site. It's catalyzed by a 5' to 3' exonuclease. Thereby, a segment of DNA, including the damaged site, is removed. In the last step is known as repair step. The enzymes are involved DNA polymerase 1 to synthesize a new stretch of DNA strand starting from the 3' hydroxyl end using the intact complementary strand as template. Finally, the newly synthesized strand is joined with the 5' end by DNA ligase to complete the repair. Another variation of excision repair is catalyzed by the enzyme glycosylase, which cleaves the N-glycosidic bond between a thymine of a thymine dimer to the sugar phosphate backbone of the DNA strand. At the next step, the phosphodiester bond is cleared by a endonuclease activity of the same enzyme which recognize a blank deoxyribose without a base attached to it. In the following step, DNA polymerase I initiates DNA synthesis at the free 3' hydroxy end, displacing the thymine dimer along with a few more adjacent nucleotides.